Welcome to the Bio Insights, where science comes alive.
Have you ever stopped to think about just how incredibly fast you are? And I don't mean your sprint speed on a track. I'm talking about the absolute lightning-fast chemistry keeping you alive right this exact second. Today, we're diving deep into how life's vital chemical reactions actually happen fast enough to keep us breathing, moving, and thinking. It's a serious question. Because without a special biological speed hack, most of the chemical reactions inside our cells would be trapped behind a massive energetic wall. They just take way too long to sustain life. We're literally talking about processes that would naturally take millions of years, somehow happening in mere milliseconds inside your cells. So how on earth does biology pull off this magic trick? Here's our agenda for today. First, we'll look at nature's ultimate speed hacks. Second, we'll tackle the energy barrier. Third, we're going inside the active site. Fourth, we've got a really fun sweater analogy for you. And finally, we'll wrap up with our key takeaways. All right, let's jump right into section 1, nature's ultimate speed hacks. Enzymes are nature's catalysts. They are these incredibly complex molecular machines that accelerate biochemical reactions with just extraordinary precision. And they don't just randomly speed things up, right? They take very specific starting materials and carefully guide them into becoming the exact products your body needs, exactly when it needs them. To really understand how they pull this off, we need to look at it from two angles: the energy changes required and the microscopic chemistry that actually makes it happen. Which brings us to section 2, the energy barrier and those tricky thermodynamic hurdles. So we have to talk about the hurdle that's standing in the way, which chemists call the free energy of activation, or delta G double dagger, if you're a fan of the math notation. Virtually all chemical reactions have to overcome this energy barrier. It's basically the massive energetic hurdle separating your starting reactants from your final products. Before a reactant can ever become a product, it has to be contorted into a highly unstable, high-energy intermediate form. We call this the transition state. Imagine a massive mountain. That intimidating peak right at the top, that's our transition state. Because of this steep energy barrier, uncatalyzed reactions are just incredibly slow. Picture a massive crowd of people standing at the bottom of this mountain. Only a tiny fraction of those molecules happen to possess enough random kinetic energy to naturally scale that massive peak and roll down the other side to actually become products. Now, contrasting an uncatalyzed reaction against a catalyzed one really reveals the sheer power of enzymes. On one hand, you have that massive energy mountain where barely any molecules make it over. But on the other hand, the enzyme steps in and provides an entirely new, alternate reaction pathway. It literally digs a tunnel right through the mountain, drastically lowering that activation energy barrier. Because the barrier is suddenly so much lower, a massive surge of molecules can flood through and react, giving us the incredibly fast rate of reaction that life absolutely demands. Now, a totally crucial point here is distinguishing what the enzyme actually alters. An enzyme absolutely accelerates the rate of the reaction by lowering that activation energy. But pay close attention to what it does not change. It does not change the starting energy of the reactants, it does not change the final energy of the products, and it definitely doesn't change the overall equilibrium of the reaction. The final destination is exactly the same, the enzyme is just providing a much faster road to get there. Okay, let's zoom in a bit for section 3, inside the active sites and its molecular chemistry. Historically, people used to describe the active site like a static lock just waiting for a specific key. But we know now that's not quite right. It's actually a highly dynamic machine. It's a 3-step process. First, the substrate drops into the active site to form what we call an enzyme-substrate, or ES complex. Then, the enzyme physically flexes and binds to stabilize that awkward transition state we talked about earlier. Finally, the chemical bonds rearrange, the transition state collapses into the final product, and the enzyme releases it, totally ready to do it all over again. To achieve this incredible feat of molecular gymnastics, the active site comes equipped with 3 distinct chemical tools. First is transition state stabilization. It acts as a flexible template to perfectly hug that fleeting high-energy intermediate. Second is general acid-base catalysis, where the enzyme uses its own amino acids to donate or accept protons, making those target bonds so much easier to break. And third is covalent catalysis, where the enzyme actually forms a temporary, transient covalent bond with the substrate to literally pull it apart before releasing the final product. Let's ground this in reality with a really fascinating example that's probably happening in your gut right now. Chymotrypsin is a digestive enzyme that breaks down proteins. What makes it so amazing is that it uses all three of these mechanisms at the exact same time. It uses a specific histidine amino acid to juggle protons for acid-base catalysis. It forms a temporary covalent bond using a serine amino acid, and it stabilizes the transition state the entire time it's doing this. It's just an absolute masterpiece of biological engineering. Time for section 4, the sweater analogy. Let's visualize this catalysis in a way we can all relate to. Listen, if you have ever tried to undress a squirmy toddler, you already understand enzyme catalysis. Our reactant is a baby wearing a sweater. The desired product is a sweater-free baby. Now, getting both of an uncooperative baby's arms extended straight up overhead is an incredibly unlikely, high-energy posture for that baby to take on its own. That right there is our massive energy barrier. But you, the parent, are the enzyme. Step 1, you grab the baby, forming the enzyme-substrate complex. Step 2, you gently force those arms up, stabilizing that super awkward transition state. And step 3, pop. The sweater comes off, the baby goes back to a comfortable low-energy state, and the product is released. You lowered the barrier? Let's bring it all home with section 5, key takeaways.
To quickly summarize our core principles today, enzymes are the ultimate biological speed hacks. They dramatically speed up life's necessary reactions by providing a low-energy alternate pathway. And they do all of this without fundamentally altering the starting materials or the final equilibrium. Instead, they rely on a highly dynamic, chemically active pocket to physically grip and stabilize that fleeting, high-energy transition state, effectively lowering the mountain so life can just race right through. We've seen how incredible these catalysts are and how they're perfectly tuned to keep the chaotic chemistry of our cells organized and rapid, which leaves us with a final, slightly provocative thought to ponder. If enzymes control the entire speed limit of life, what exactly happens to us when these molecular machines break down, mutate, or go into overdrive? Well, that is a fascinating topic for another time. Thanks for joining me for this explainer. Keep asking questions, and I'll catch you on the next one.